Upon completion of this module, you should be able to differentiate prokaryotes and eukaryotes protein synthesis. Protein synthesis in the cells is done during translation, which is the second step in genetic expression. During translation, ribosomes decode information contained in the mRNA to proteins and is a universal process occurring in both prokaryotes and eukaryotes. Fundamentally, the process is the same in both prokaryotes and eukaryotes, but there are also some differences. We will look at both similarities and differences, starting with the similarities. Let us see the similarities between prokaryotes and eukaryotes. Both use an mRNA template. mRNA is synthesized from DNA. Protein synthesis is done via ribosomes. All 61 codons are similar in both groups. All 20 amino acids are the same in both groups. Protein synthesis occurs in the cytoplasm. The translation is completed in 4 stages. They are initiation, elongation, translocation, and termination. Formation of peptide bonds. tRNA picks up and delivers the correct amino acids. rRNA molecules of a large subunit of ribosomes are where peptidyl transferase activity is accomplished. Proteins have post-transitional modifications. Now we will see the differences between prokaryotes and eukaryotes. In prokaryotes, transcription and translation are continuous processes that occur simultaneously in the cytoplasm, whereas eukaryotes, transcription and translation are separate processes. Transcription occurs in the nucleus and translation in the cytoplasm. 5' end of the mRNA is used for translation immediately in prokaryotes, but in eukaryotes, the primary transcript is processed after transcription, then transported to the cytoplasm where ribosomes initiate translation. Prokaryotic ribosomes consist of the 30S and 50S, the smaller unit and larger unit respectively, whereas eukaryotic ribosomes have smaller subunit and larger subunit as 40S and 60S respectively. Prokaryotic ribosomes consist of 21 proteins and 36 proteins, the smaller unit and the larger unit respectively, whereas eukaryotic ribosomes have smaller subunit and larger subunit as 36 proteins and 49 proteins respectively. Since there are no membrane-bound organelles in prokaryotes, the rhizomes float free in the cytosol, whereas eukaryotes have an endoplasmic reticulum. Therefore, proteins synthesized in ribosomes are attached to the ER. Prokaryotic mRNA is polycistronic, but eukaryotic mRNA is monocistronic. In prokaryotes, mRNA operates as the template for the synthesis of many polypeptides, but mRNA acts as the template for a single polypeptide in eukaryotes. Only one type of translation initiation system, cap-independent in prokaryotes, whereas in eukaryotes, 2 kinds of translation initiation systems, cap-dependent and cap-independent. Prokaryotes have many start sites and Shine-Dalgarno sequences along with mRNA, and have 8 nucleotides up from the start codon. The SD sequence acts as the ribosome binding site, but it is absent in eukaryotes. Eukaryotes has only one start site located near the 5' end region of the mRNA. Kozak sequence is absent in the prokaryotic mRNA, whereas it is present in the eukaryotic mRNA. In prokaryotes, the first amino acid in protein synthesis is formulated, but in eukaryotes, no formulation of the first amino acid. No poly-A tail is added to the mRNA in prokaryotes. In the case of eukaryotes, a poly-A tail of around 200 adenine nucleotides is added to the 3' end of the mRNA. Since there are no introns in the prokaryotic mRNA, processing is not required, However, in the eukaryotes, mRNA has introns and exons and processing is required.